Well, aloha everyone, and welcome to the lecture on cellular organization for Anatomy and Physiology 1. To introduce what we're going to discuss this chapter, we're going to be talking about all of the different parts within a cell, including the importance of the plasma membrane, which surrounds the cell and keeps the inside separate from the outside. The inside is the cytoplasm, and we'll talk about the components of the cytoplasm. And then last but not least, we will talk about both mitosis and meiosis. So that's cellular division for normal cells, say when you cut yourself, and then also that's mitosis. And then also the type of cell division that occurs when we are creating sex cells or sperm and egg. And then last but not least, we will talk about the effects of aging on the cell. Just to introduce you a little bit, we have three major parts of the cell. The first is the plasma membrane, which surrounds the entire cell and again keeps the inside separate from the outside. Within the cell, we have the cytoplasm, and the cytoplasm can be separated into the cytosol, which is the liquid component, and then the organelles, which are membrane-bound, basically miniature organs of the cell that each have their own specific functions in order to keep the cell running. The nucleus is going to be the largest organelle within the cell. It's going to contain chromosomes and all the genetic material necessary for the cell to run. So this is where the DNA is going to be housed. You can picture this like kind of the boss's office. Now I know this is a very overwhelming image, but don't worry, we're going to walk you through every single one of these one by one. This here is an animal cell, and there's a couple of ways that we know that, just to talk you through the differences between plant and animal cells. So animal cells are going to be the only type of cells that have this flagellum, or cilia, either one of those. Additionally, you don't see any chloroplasts, which are specific to plant cells. You also see that we do have a cell membrane, but not a cell wall. Now, all plant cells have cell walls, so that's going to be a pretty quick and easy way for you to determine whether or not you're looking at a plant cell or an animal cell. Now, most of the rest of the organelles are going to be shared between plant and animal cells. So this is the nucleus that I was talking about previously. This is the boss's office. This is where all of the information is housed. So as expected, inside here we have the chromatin. Now, chromatin is your DNA before it is condensed into what you think of when you think of chromosomes. You think of those pretty little X's. That's condensed DNA, and we will talk about that when we get to cell division. But for now, the cell is actually actively using this information, and so it's going to be decondensed into what we call chromatin. From there, that DNA is going to create RNA, and RNA is going to be allowed out of the nucleus. Now remember, DNA is not. So RNA is going to be allowed out of the nucleus through these nuclear pores. You can see them depicted here as these little red donut-shaped holes. And then once it leaves the nuclear pores, it's going to attach to these things here, ribosomes. We also see ribosomes that are connected to this structure here. This is the endoplasmic reticulum, also known as the ER. So again, RNA is going to come out of the nuclear pore. It's going to attach itself to a ribosome, and once the ribosome is attached, it's going to start the process of protein synthesis. But I digress. We'll get to that in a little bit. The cytoplasm is what is defined as the cytosol plus all of the organelles except the nucleus. So that's going to be everything in here besides the nucleus. And the cytosol, again, is that fluid portion, the liquid that is inside here. Remember, that's all going to be water-based. So it's a water-based liquid. But there's a lot of other things in there, too. It is not water. It actually has salts and ions, etc. Now, as I mentioned that this is the rough ER, this is where the ribosomes are attached, we also have a smooth ER. And that's here, and it's just depicted as without ribosomes, which is true, but that's how they saw it when they looked under the transmission microscope, as they saw these ribosomes. That part looked rough, this part looked smooth, so that's what they called it, the rough and the smooth ER. And you'll notice that all of these organelles are membrane-bound, that's what's depicted here, and they're all interconnected. So you can see that the endoplasmic reticulum is directly connected to the nucleus, which is directly connected to the smooth endoplasmic reticulum. So all of these organelles actually are going to have an interplay amongst them. That's this membranous system. Now, this here is the Golgi apparatus, or the Golgi complex. After proteins are created, they're created on the ribosomes, either free ribosomes in solution or ribosomes that are bound on the rough ER, and then they're going to go to a protein sorting factory, and that's this rough endoplasmic reticulum here, the rough ER, and we'll talk a little bit more about how the proteins are sorted when we give the rough ER its own slide. Let's see, what else can we discuss here? This here is the mitochondria. This is the battery of the cell, basically. It's where we're going to be creating ATP. Mitochondria and chloroplasts, incidentally, although you won't see any here because it's an animal cell. But both of these are considered to be originally their own organism, much smaller prokaryotic organism, that was then engulfed by a eukaryotic cell, and then instead of being digested the eukaryotic cell noticed that it actually has a creative way of creating energy. And so they keep it around and allow it to create ATP for them, which is that energy carrier molecule. Again, that's the mitochondria depicted here. Um, Peroxisomes and lysosomes are the cellular trash cans, basically. These are going to secrete peroxide, or lysosomes are going to help digest and recycle materials. These are the cellular waste and or recycling containers, whereby they're going to help deal with anything that might be misfolded protein / anything that might be a foreign invader. These here are centrioles. Centrioles are going to be very important when it comes to cellular division, but for most of the time they're just going to hang out in the cell, but during cell division they're going to have one set on each side, and they're going to create spindle fibers that then attach to the chromosomes and pull them to opposite sides. Again, you'll see that just a little bit later in the lecture. Here are some specialized type of region. Here's the microvilli. We'll talk about this extensively when we talk about something like the intestines, for example. So these are regions where they've increased the surface area so that you can have more exchange of nutrients or waste products across that membrane. And although I didn't touch on it much, the cytoskeleton is very important. So the cytoskeleton is found here. We also have intermediates of the cytoskeleton here. It's basically all of the structure and scaffolding. Here's microfilaments. We'll talk about them individually later, but all of these are going to be scaffolding parts that are going to help keep the cell structure sound. All right, so again, I've talked about the different regions now. We've talked about the plasma membrane. Talk about the cytoplasm, which again contains the cytosol, or the liquid portion, and the organelles, and then we'll talk about the nucleus. So first, let's focus on the plasma membrane. So plasma membrane is very unique. You might remember when we talked about biomolecules that it's composed of these phospholipids, and the phospholipids are what we call amphipathic. So they have a region that is both water-loving and water-fearing, and they arrange in such a way that the entire water-fearing layer is on the inside and the water-loving layer is on the outside. And so this part is able to interact with an aqueous environment. This part is also able to interact with an aqueous environment because it's a double phospholipid bilayer. But the inside region, where we have the two sets of phospholipid tails oriented towards each other, is actually going to be hydrophobic. So it's not going to let water through. So that's called the lipid bilayer. And it's selectively permeable in that certain things are going to be allowed through and other things are not going to be allowed through. It's a very sturdy barrier that's going to house the cytoplasm on the inside, and the outside is going to have what we call extracellular fluid. And these are going to be both fluid but have different compositions. So we want to have what we need inside the cell and export the cellular waste outside of the cell. And again, depending on what we have inside the cell, we might need to draw something in if we have too little of it, or secrete it out if we have too much of it, so that we can maintain the word homeostasis comes to mind, right? You'll hear that a lot throughout the semester. But let me talk about what's plugged into this plasma membrane here. So this guy's here, these are cholesterol. Cholesterol is going to help with the stability and flexibility of the plasma membrane. We also have these specific types of proteins that are going to stick out of here. These are glycolipids. We have carbohydrates that are going to interact here with that lipid region, and those are actually going to be, and we have another set here, these are going to be types of proteins that are going to be identifying markers that are going to say the type of cell within the body, for example, or what specifically which person this comes from. You can actually have identifying markers that say this is Dr. Griffith's cell, for example. These are other types of proteins. We have different proteins that are going to go all the way through the plasma membrane. These are called integral proteins or transmembrane proteins. Sometimes they form these pores, like these channel proteins come together and form a pore. And then sometimes they're going to form these just multi-passes that are going to have different information relayed from the outside to the inside. So something that's bound here in the outside will send information to the inside and cause an intracellular cascade. So the plasma membrane is very important. It not only separates the inside from the outside, but it also is going to help relay information from the outside to the inside and bring in the nutrients and secrete the waste. So it's going to be a very important barrier that's semi-permeable. Again, we have multiple different types of membrane proteins. The two major ones are going to be integral. We just talked about those. Those are transmembrane proteins. They're going to be the ones that go all the way through. And then we have peripheral proteins. And peripheral proteins are only going to be embedded, say, on one side or the other. Now, there's a ton of different functions of these membrane proteins. And I'm going to talk about each of these individually in just a second. But we can have ion channels, carrier proteins, receptors, enzymes, linker proteins, and identification proteins. So let's talk about these one at a time. So ion channels are going to basically make a pore like this, in which ions are going to be able to get through from one side of the membrane to the other. And oftentimes plasma membranes have specific channels for multiple different common ions. And we may have more or less of these channels in particular regions that may need these ions more than others. So for example, the heart may have a different amount of ion channels than, say, the eye cells might. However, all of them are going to have different channels that are going to allow, again, ions to pass into the cell. Now, some things are going to be a little bit larger. So if you can picture, ions are pretty small. Sometimes we might have something that's a little bit larger that needs to be carried through. And we'd have these carrier proteins. You can see carrier proteins have a conformational shape whereby they're going to face either the outside or the inside. And then once they pick up their substance, in this case it's coming inside, so it'll be facing the outside to begin with. It'll be picked up, and then the conformation will change such that that protein or whatever it is that's going to be transported will be allowed within the cell. And these carrier proteins are also known as transport proteins or transporters. Now, we have specialized type of receptors that are on the outside of the cell as well. These are receptors, proteins that are going to be specific for a ligand. So you can picture this like a lock and key model. Now, the ligand's not going to enter the cell, but if the ligand binds to the receptor, there's going to be an intracellular change on the other side of that receptor that's going to alter the cell's function, upregulate something, downregulate something, right? And a good example of that is ADH, or antidiuretic hormone, and that's going to bind to receptors that are specialized to the kidneys. That's going to help change the water permeability of plasma membranes to allow more water in or less water in, depending on the osmolality, or the amount of water, free water in solution in the blood, for example. This one here is depicted as an enzyme protein. Now, enzymes are going to take substrates and turn them into products. In this case, this is a catabolic reaction because it's taking a larger substrate and breaking it down into smaller products. But there's going to be an equal and opposite enzyme that takes these two and puts them together to make that larger barbell as well. And these enzymes are going to be on the peripheral region of the cell, again, embedded in the plasma membrane. And they can catalyze a reaction either inside or outside, depending wherever the active site is. So a good example of this is lactase. Now remember, anytime you see ACE, we're talking about an enzyme. Anytime you see OS, we're talking about a sugar. And if lactase is protruding on the outside of your epithelial cells inside your intestines, that's going to help break up the disaccharide bond in lactose so that you're able to digest that lactose. And people who are lactose intolerant generally aren't able to create the enzyme lactase. And this is why they might take something like lactate. It's basically an exogenous or an external way of consuming lactase so that then you are able to digest lactose. Another type of membrane protein here is a linker protein. Here we have an integral one or a peripheral one. They can either be all the way through or they can be on the outside, but basically these are going to be anchors. It's going to help with the scaffolding. They're generally going to connect to the inside as well as to the extracellular matrix, and that's going to help provide structural stability for the cell and also help it maintain its shape. Sometimes it'll actually help two cells link together with, from one to another, and that will help make something like a water impermeable barrier or something like that. So you can picture the way that your skin cells, if you were to just pinch the back of your hand and try to pull, you're not going to separate those cells because they're held together by linker proteins. And on the outside, we also have these glycoproteins. Now, glycoproteins are basically identity markers. They're going to distinguish one cell from another cell. Not just distinguishing a heart cell from a heart cell, or a heart cell from a brain cell, for example, but also distinguishing my heart cell from someone else's heart cell. And this is the reason why people who get heart transplants or any organ transplants end up having to take immunorejection drugs or anti-immunorejection drugs for the rest of their lifespan, so that they have a reduction in the ability of these identity markers to interact with the external environment, because it could cause a reaction. Okay. So membranes are fluid. So we think of it as, you look at all those little phospholipids and you think maybe that they're solid, but actually it's a phospholipid bilayer is more of a fluid mosaic. It's got all of these different phospholipids touching one another, but they're not actually anchored to one another. And so they're able to move easily amongst themselves. And that's going to allow the proteins that are in that cell to, like, inside the cell membrane, to kind of aggregate however they choose. So these proteins are mobile on one half or the other. They're not going to be able to switch from the outside to the inside, for example, but they are able to move about amongst the membrane. And same thing with cholesterol. Cholesterol leave these little plugs, and they're able to move about within the membrane as well. Usually they try to find equilibrium, and they're basically going to serve to help stabilize the membrane and help it be a little bit less fluid, because they're definitely going to be less fluid than, say, the phospholipids. But the most important part about the plasma membrane is that it is selectively permeable, and that means that it's going to allow certain things in and keep certain things out. So you can picture it like the gatekeeper of a large wall around the city And that way it's going to help decide whether or not certain things are needed within the city or certain things are not needed within the city. And so they are going to make the decision based on the concentration inside and outside of certain molecules. Now, some things are able to make it through all the time. So it's always permeable to certain molecules that are small molecules, nonpolar molecules, uncharged molecules. They'll be able to slip between the cracks in the fence, for example. But larger molecules are going to have to be brought into the cell. And we use transmembrane proteins for that, again, channels or carrier proteins, to help increase the permeability of the membrane to whatever it is we want to bring in. But certain things that are like macromolecules, much larger, are only able to be brought in by vesicular transport. And we'll talk about vesicles or vesicular transport in just a second, but basically the entire, what we call endomembrane system, is going to be linked by these vesicles that are going to be secreted from one organelle to the next, etc. But first, let's step back for a second and talk about gradients. So I just talked about based on concentration, right? And what I mean by that is the difference in concentration from one side of the membrane to the other side of the membrane. And we're specifically talking about chemical A or chemical B, right? A concentration gradient is going to be specific to sugars. So we talk about the sugar gradient, or salts, the salt gradient would be separate. And there's not necessarily going to be any correlation between the two. So you might be higher of one thing on the inside of the cell than the outside, and something else could be higher on the outside than on the inside. Anyway, concentration gradient is when we have a difference, when it's not equal from whatever that chemical that we're looking at is between one side and the other side. And we can also have a gradient in terms of electricity, and that would happen if we had ions. Remember, ions are going to be charged versions of the atoms, and if the ions are charged and we have a higher amount of positive ions on one side than the other, then we can end up with what's called an electrical gradient. And this is going to be very important when we talk about the nervous tissue, for example, or heart tissue, which is reliant on that nervous tissue to fire. And the combination of both the concentration gradient and the electrical gradient is called the electrochemical gradient. And this is what everything in the cell is going to run on, basically. And in order to maintain these gradients, so that we're able to maintain, again, homeostasis, we have to be able to allow things in or out. Again, it's going to be selectively permeable. And so we have to have transport processes that move things across the cell membrane. And we have two different ways in which things are going to be moved: the passive process and the active process. And I like to make this akin to, do I have to spend money or is it going to be free? So a passive process would be a process where we're not going to spend any money. Again, money in this version is going to be ATP or other types of cellular currency. And an active process is going to involve energy of some sort. So a passive process would be something like simple diffusion. Things are going to go from a concentration of high concentration to low concentration on their own. If they're able to go across that barrier, no problem. If they need to be assisted across that barrier, but they are still going from high concentration to low concentration, and they go through something like a carrier protein or a channel protein, that's facilitated diffusion. And if we're talking about simple diffusion, just basic high concentration to low concentration directly across the cell membrane, but we're talking about water, then we're talking about osmosis. Again, osmosis is specific to water. So when someone says, I'm going to sleep on my anatomy textbook and learn all the material by osmosis, not only is it not going to work, because that's ludicrous, but it also isn't using the language properly. Okay, so active transport or active processes are going to be when we are actively using energy to move something from one side of the gradient to the other. And usually when we are doing this, we're going against a concentration gradient. What I mean by that, if we're going with the concentration gradient, we're going from high to low, right? And that's easy. That's going to be without any energy required. But if we're going from low to high, like we want to put whatever substance it is outside of the cell, but there's more of it on the outside than on the inside concentration-wise, then we're going to need to actively transport it out. And then last but not least, we can use those vesicles that I was talking about to transport as well. This will be for the larger macromolecules that I was discussing previously. So let's talk about the passive processes. So passive processes include something like simple diffusion. And diffusion is basically when we have a concentration gradient and then over time we reach equilibrium. All right, so here we have we're depicting it in terms of dye. So we have dye at the bottom of this beaker and no dye at the top. And over time, remember, molecules are bouncing off of one another. And the higher the temperature, the faster they're bouncing. So the faster this rate will occur. So as you increase, picture boiling water is going to have more motion than colder water. And so as we increase the temperature, we're going to also increase the rate of diffusion. So there's multiple different things that are going to affect how quickly we're going to go from this gradient, which is really highly concentrated here, lowly concentrated there, to equilibrium, which means that both sides, or whatever the substance is equally distributed. But if we're talking about a membrane, that means that both sides are going to have equal amount, equal concentration. The mass of the substance also matters, and there's a short little excerpt that we'll talk about when we get to the lab, where we put two different substances inside little holes in the agar, and the larger substances are going to take longer to diffuse because they're a little bit heavier, and it might take them a little bit longer to get through holes, or in this case to travel through the water. And also the surface area, in this case we're talking about the cell, right? And that's why we try to keep our cells small. That's why we divide our cells and don't just become one large unicellular organism, but instead choose to become multicellular organisms. Again, I say choose as though we have a choice. And last but not least, the distance across which we have to diffuse. So if this were a much longer tube, it would take a lot longer to reach equilibrium than if it were the size of the tube that it is. All right, so again, diffusion is just when something is going from a high concentration to a low concentration until it reaches equilibrium, and it does that without any sort of energy involved. Now, facilitated diffusion is going to be, again, from high concentration to low concentration, but it's going to be for things that are either too charged or too polar to get through that lipid bilayer, and so we're going to need either channel proteins or carrier proteins. And channel proteins, again, are just going to basically be a pore, and a carrier is going to be more along the lines of having one face to the outside that has a different shape, picks up that molecule, and then conformational change happens so that it's pulled inside and delivered to the inside of the cell, or delivered from inside to outside. But either way, the carrier is going to change shape to deliver one at a time instead of being just a pore through which something would come. All right, so this is facilitated diffusion. This is an example here. Here's a channel protein. Now, as I mentioned, the channel protein is going to pull things from one side to the other. So here we have potassium. Potassium here is in the cytosol of the cell. The extracellular fluid is depicted here in blue, and it's going to travel through the channel protein, then through the pore, out into the extracellular matrix. And sometimes the gate's going to be open, and sometimes the gate's going to be closed. So sometimes it's going to just go right on through because it's open, and sometimes it's not going to be able to. But either way, we're not seeing a conformational change in the actual part of the protein that's going to be the channel. We're only going to see it in like this gate region. Now, a carrier protein is going to have a conformational change from the inside to the outside repeatedly. You can see here it's facing the external environment. In this case, we're looking at glucose. Again, it's a hexagon. That glucose molecule gets picked up. The glucose transporter changes shape and pops it inside the cell. Again, we are still going from a high concentration to a low concentration. So it's still considered diffusion, but in this case, because we have to have help, it's considered facilitated diffusion. All right, so this is showing three major types of diffusion. We have simple diffusion, whereby it's just going to be able, it's a small, non-charged molecule. It's going to be able to go right on through, or in the water as well is able to make it through, even though it is polar, by simple diffusion. Or it can go in by a channel, or it can go in by a carrier. None of these are going to require energy, though, however, because of the fact that they are coming from a high concentration to a low concentration, and because we're going with the concentration gradient, we're not going to need to use any cellular energy. Now, osmosis is what we were just talking about in terms of a small molecule being able to move by diffusion, but specifically to water. And osmosis is talking about the net movement of a solvent from a selectively permeable membrane from an area of high concentration to an area of low concentration. So again, we are going with the concentration gradient, but we are only going to be talking about the motion of water. All right? Now, if we are talking about the concentration of water on the inside or the outside, really we could be talking about the concentration of anything, but for most of these we're going to be talking about the concentration of water. If we are equal, it's what we call isotonic. So isotonic means that the net flow of water in to the cell and out of the cell is the same. Now, if the net flow of water into the cell is larger than the net flow of water out of the cell, then we're going to end up undergoing what we call hemolysis, and that's because we're looking at a red blood cell. Lysis just means broken, and hemo being red blood cell broken. But lysis of the cell, because too much water goes in, not enough water goes out, and it bursts. And this happens if we are in what's called hypotonic solution. That means that we have a higher concentration of water on the outside than on the inside, and it's just going to flow right on in. Now, if we are in what's called hypertonic solution, that means we have a higher concentration of water on the inside of the cell than on the outside of the cell. We're trying to reach equilibrium, so we're going to have more water coming out than water going in. Then we're going to undergo what's called crenation, or basically the cell folding in on itself because all of the water has been lost. So tonicity of the outside environment is very important, right? You want to make sure that we have the same tonicity on the inside as the outside, even though we're going to have different concentrations of ions. The overall concentration of water should be approximately the same so that we can maintain our cell's integrity. All right, so let's talk about active processes. So we just talked about diffusion and different types of transport that didn't require energy. Now we're going to talk about active transport, which does require energy. Now, the energy that's created when ATP is created is going to be stored, basically. So ATP is a cellular carrier molecule for energy, and that energy is stored in that high energy bond between the second and third phosphates, and when it breaks, it goes from the high energy form, which is depicted as ATP here with the little points, down to the low energy form, which is ADP, right? So when we go from tri to diphosphate, we release that phosphate. When we do that, the energy expenditure is going to allow something to change shape. All right, so the reason that we need to use energy in the first place is because we are going against a concentration gradient. So we have more sodium on the outside than we do on the inside, right? But we want to put that sodium outside, even though we already have more on the outside. So we're going against the concentration gradient. So we're going to spit these 3 sodium out when we are using the conversion of TP to DP and freeing that free phosphate and getting that energy from there. So we're actually going to be going against the concentration gradient by expending energy. And this pump is actually in concert with a potassium pump, whereby as soon as we expel those 3 sodium, we're also going to bring in 2 potassium, and that's going to free up the phosphate so that we can then put it back into another ATP molecule, and again another ATP molecule can come over here, release its phosphate, and start this whole process over again. So basically anytime that ATP is involved in the process at all, and it's pumping something from a low concentration gradient to a high concentration gradient, then it's going to be called active transport. Secondary active transport is going to be any time that we are driving substances against their concentration gradient, as I mentioned previously, and we can do that by kind of tricking the system. So sodium is often used to go back and forth and back and forth. And you could picture this, let's go back to the gatekeeper analogy, where the gatekeeper says, hey, doctors can come in, but no lawyers can come in. But if a doctor comes up paired with a lawyer, then the doctor and the lawyer can both come in. And that's what happens here. This is a symporter. In this case, sodium is going to come in with glucose, or sodium in with an amino acid. So it's going to allow something in that wouldn't be allowed in on its own. And originally, in order to get a higher concentration gradient, we have to export it out of the cytosol. So here we have, again, sodium coming in with calcium going out, sodium going in with hydrogen going out. These are called antiporters when we're going to go opposite directions. So sodium can go one way or the other, and it can either bring something with it, or as it's traveling in, it can send something out, and vice versa. Okay, so anytime that things are going in the same direction as whatever is also coming in, that's a symporter. If they're going in opposite directions, one coming in and one going out, that's an antiporter. And this is a type of secondary active transport whereby we can use the energy from one of the concentration gradients that's working to its advantage, and we pull something else along with it against its own concentration gradient. Right? And in this case, it doesn't necessarily cost us cellular energy in the form of ATP, because the energy that's stored is actually going to be coming from the transport of another molecule that is already leaving the cell. Okay, so let's talk about vesicles for a second. So vesicles are a way by which we can transport macromolecules from one location within the cell to another location within the cell to the external environment. All right? So we have a specialized type of a vesicular transport called endocytosis, and another type called exocytosis. Endocytosis is when we are bringing something in, so endo, enter. Exocytosis, something is exiting the cell, so exit, exo. So anyway, for endocytosis, where we are going to be using receptors to pull something in this case we are bringing in these little LDLs, and we're going to bring them in by way of a clathrin-coated pit. And clathrin is basically going to have a small conformational change until it forms what we call this micellular structure, which is going to be the vesicle. And the vesicle is going to be coated on the outside by clathrin. Eventually that clathrin is going to be taken off, that's uncoating, and then it'll go back up to the outside, and it's just a specific protein that helps basically create the vesicular shape. So it's going to help with the invagination here. Anyway, it's not needed after the vesicle is made, so it's uncoated, and then that uncoated vesicle is going to fuse with, here we have an endosome. And if it's something that we wanted to digest, it would end up being degraded into the lysosome. If it's something that we wanted to use or recycle, we might put our own cellular tag on it and change it a little bit, and then recycle it back out to wherever it needs to go. In this case, these are recycling of the receptors that we use to pull in LDL. Okay, so active transport is basically a way by which we can pull things in against a concentration gradient. In this case, we're using vesicles because these are very large molecules that are not going to be able to go in on their own. This is phagocytosis. It's a very specialized type of vesicular transport. It's kind of like a digestion. It is a digestion of something that is an invader. In this case, it's a microbe. So we have these specialized receptors on the surface of the cell that say, hey, this is a microbe. The microbe gets bound. And then we have these regions here that are called pseudopods. Pseudopods reach out and invaginate around the microbe, pull that microbe in. That's going to be in a region called a phagosome. Phagosomes are for digestion of something that is, in this case, a foreign invader. The lysosome is going to fuse with the phagosome. The lysosome has the digestive enzymes in there, and then that's going to help digest whatever it is into the components that it's created of. And we're going to make some of them cellular waste, and anything we're able to recycle, the cell is going to recycle and then send out to wherever it's going to be used within the cell. And this is what that looks like in an actual image. Here's a microbe being destroyed. Here's the pseudopods reaching out, being pulled into the white blood cell, and then being broken down in little pieces by the white blood cell, which we'll talk much more about white blood cells when we get to immunity. All right, so if phagocytosis that we just talked about was cell eating of larger things, pinocytosis is going to be something like cell sipping, right? Taking in small little fluid sacs that also have dissolved solutes in them, and then again fusing them in the same way with the lysosome, digesting them, and then releasing the digested solutes that are able to be used into solution, and then taking whatever we can't use and secreting it into cellular waste. All right, so again, endocytosis is when we are bringing something in. Exocytosis is when we are using vesicles to send something out. So exocytosis is still membrane-bound vesicles, but instead of coming in, they're now fusing from the inside with the plasma membrane and then basically popping open like bubbles to release their contents into the extracellular fluid, which is the fluid on the outside of the cell. Now, both endocytosis and exocytosis are basically like these constant in and out, and that's called transcytosis, is the combination of all of the endocytosis and exocytosis that the cells use to be able to transport substances from one side to the other side. All right, so this is an overview of all of the things that we just talked about. So we have active transport processes and passive transport processes. The first ones we talked about were the passive processes, such as diffusion, right? Diffusion is when we are going down a concentration gradient. All of these are going to be along the concentration gradient, from a high to a low until we reach equilibrium. And that's what we talked about. We talked about the dye example in the beaker. Simple diffusion is basically when we are going to have the motion of a substance down the concentration gradient without the help of any transport proteins. But if we do need to have transport proteins, remember we had carrier proteins and channel proteins, and that's called facilitated diffusion. Again, we're still going with the concentration gradient from high to low, but in this case we're going to need some sort of carrier, and that's if we have a polar substance or a charged substance, things like sugars, which are pretty large, different types of vitamins and ions, which might be charged. Osmosis is when we're talking about the passive movement of water, but we're, again, only talking about water, and we are talking about going from a high concentration to a low concentration until equilibrium is reached. So it's basically diffusion, simple diffusion, but still specific to the diffusion of water. And in this case, again, the solvent is going to be water only in living systems. Now we have active processes. We have a couple of different types of active processes. Active transport is when we are expending energy, usually in the form of ATP being broken down into ADP, and that's going to allow us to move some substance across the membrane against the concentration gradient. Again, normally where we'd be going from high to low, that's what it wants to do. So against that gradient would be from low to high, and it would require energy to do that. Primary active transport is when we are moving things, again, against the concentration gradient. We're going to use pumps to do that are going to be supplied either by ATP, or another thing I didn't mention is by hydrolysis. So sometimes hydrolysis is actually going to be the way that we are breaking that phosphate group off of the ATP. So hydrolysis would be breaking down ATP into ADP and free phosphate, which is what is going to actually be releasing the energy that we need to drive the primary active transport pumps. Secondary active transport is basically when we're hitching a ride, right? It's a coupled active transport system. So one is going to be coming in that is going to be going with the concentration gradient, and something else is going to be either coming along with it or leaving at the same time. We have symporters whereby two things come in at once, and antiporters whereby one thing comes in and one thing goes out, again, opposite directions. Now, we talked about vesicular transport. We talked about endocytosis when we move things in, exocytosis when we moved things out. Endocytosis, we had receptors that were mediated on the outside. We talked about those clathrin-coated pits. LDL was the one that we use as an example, but there's several other things that are being brought in by those receptors. And the ligands that can interact with the different types of receptors can include things like transferrin, again, the LDLs that we talked about, vitamins, specialized hormones, and antibodies. And antibodies are going to be very important when we talk again about our immune system and immune function. Phagocytosis, speaking of immune function, is cell eating. This is going to be very important when we are engulfing things like bacteria, but also involved in the destruction and digestion of dead cells of the surrounding tissue so that we can take what we think is usable and recycle it into the cell before we expel anything that is not going to be usable into the extracellular fluid. And then we also had pinocytosis, which is when we are moving a fluid in, and inside that fluid we also have different things that we might need, the different solutes, and the solutes will come in from the extracellular fluid, and then same as the phagocytosis, they'll end up interacting with those lysosomes and then either be broken down or recycled or secreted as waste. Exocytosis is when we are using vesicles to secrete something. Again, those are the bubbles that are meeting the inside of the plasma membrane and bursting open. Some examples of those are things like neurotransmitters and hormones that might be secreted, and also digestive enzymes that are going to be secreted in specific cells and also during specific times, right? You don't want to always have digestive enzymes. You only want digestive enzymes after you've eaten and there is something to actually break down. And the whole of that, right, endo and exocytosis, is called transcytosis. And a lot of things are going to move into and out of multiple different sets of cells. So a lot of things are going to be passing back and forth, for example, from blood plasma into interstitial fluid, into the cells, back into the interstitial fluid, back into the blood plasma. So transcytosis is the entirety of that using vesicles. All right, so I know I've touched on a lot here. Here we're going to talk about all of the different parts of the cell again, focusing specifically on the cytoplasm for now. So the cytoplasm has a liquid portion and a membrane-bound portion. And the cytosol is the liquid portion. It's the intracellular fluid portion of the cytoplasm. And as you remember previously, we talked about organelles as mini organs within the cell. So they're tiny organs, basically. They're specialized membrane-bound structures inside the cell that have specific functions, and again, form defines function for all of biology, so their specific shapes are going to help allow them to perform those specific functions. All right, so we talked about the plasma membrane and how it was a fluid mosaic, a lipid bilayer, had lots of different proteins inside there, and was going to help surround the cell to be able to separate the inside from the outside. Again, the inside being the cytoplasm and the outside being the extracellular fluid. And the function of the plasma membrane is protection, mainly, but also transport of things in and out. Receptors are there to be able to identify the cell. Again, cell identity markers, linker proteins, and mainly mediating things into and out of the cell to help maintain homeostasis. Now, the cytoplasm, again, that's going to be both the cytosol, which is the liquid portion, and all of the different organelles, is also going to be very important because this is the site of all of the intracellular activities, with the exception of things that are occurring in the nucleus. And specialized functions happen in the nucleus that don't occur anywhere else. But most of the intracellular activities are actually going to happen in the cytoplasm. So this is kind of like the factory floor if the nucleus is the boss's office. Again, the cytosol is comprised of two different things. The cytoplasm is composed of two different things, the cytosol and the organelles. The cytosol is water soluble and has a lot of different particles and solutes inside there, as well as glycogen. And inside we also have the cytoskeleton. So I forgot to mention that. The cytoskeleton is basically a scaffolding network. And it has three major protein filaments, microfilaments, which are the smallest, intermediate filaments, and the microtubules, and that's involved in maintaining shape and organization of the cell contents, and also it's responsible for the cells moving around within the body. And that sounds kind of crazy for now, but if you realize that a lot of cells are actually constantly in motion with the cells around them, it's actually not as far-fetched as you think. All right, so what type of organelles are we going to talk about? Well, I've touched on many of them in that introduction, but they're each going to get their own due. But organelles are specialized membrane-bound structures that have specific shapes and specific functions. Now, the centrosome we actually don't use very often until we get to microtubule function, which is either for motion in specialized types of cells or for mitosis. So the spindle fibers are going to come from either side of the centrosomes, and that's going to pull, that's what I mean by motion, pull the chromosomes to opposite sides prior to the splitting off of the cell into two cells. Cilia and Flagella are specialized type of motile projections. Now, cilia are more like oars on a rowboat, and flagella is more like an outboard motor. So cilia, there are many of them, and flagella is going to be single, generally just one. What comes to mind, obviously, right off the bat is a sperm cell. And I also want to point out a major difference between cilia and flagella. So cilia are generally found lining regions that have liquid going across them. Right, they're found in the uterus, for example, and in the airways and the trachea, right? And cilia are involved in moving fluids over the cell's surface. So cilia stay put and the fluids migrate over it, whereas flagella are meant to move an entire cell, like a sperm cell that needs propulsion from one side to the other. So that's another major difference between cilia and flagella. Another thing we'll talk about is the ribosomes. We're going to talk about these extensively. Ribosomes are involved in protein synthesis, and they are either free in the cytoplasm or attached to a region of the endoplasmic reticulum known as the rough ER, simply because it appears rough because of all of these ribosomes that are attached. Ribosomes have two different subunits, one large and one small. We'll get to that in just a minute. And the ribosomal RNA is going to bind to an mRNA transcript that is going to basically be the blueprint that's going to allow the creation of a protein. It's going to be able to use over and over and over to create that protein as many times as possible before it's degraded, and the ribosomes are going to assist in this. All right, so the endoplasmic reticulum, again, it's in the endomembrane system. It's part of the endomembrane system connected to the nucleus and also to the smooth ER. The rough ER is called the rough ER because it's covered in ribosomes. The smooth ER does not have ribosomes on it, so it just looks a lot smoother. And they do have two separate purposes. So the rough ER is going to be for the synthesis of proteins, basically. It creates glycoproteins, it creates phospholipids, it creates a lot of different types of biomolecules that are going to be either transferred from one organelle to another, or end up inserted in the plasma membrane, or secreted during exocytosis, whereas the smooth ER is going to be responsible for synthesizing fatty acids and steroids. It's also going to be involved in drug detoxification and storage and release of calcium ions in muscle cells specifically. The Golgi apparatus, as I mentioned again, it's also part of that endomembrane system, but it's not connected directly to the ER. So proteins that are created in the ER are going to end up in small little vesicles that are going to then fuse with the Golgi complex. The Golgi complex has multiple different membranes that are called cisternae, and they're going to be divided into whether or not they're cis, which is the face, the medial, which is the middle, or the trans, which is the exit, right? So either it's coming in, the entry, or the medial, or the exit. And as the proteins go through these different sets, they're going to get different identifying markers that make them more and more ready for transport. We'll talk about that specifically later on, but if you were thinking about something like the U.S. Postal Service, right, when someone picks up an envelope, first they have to decide, is it going to the West Coast or to the East Coast or to Middle America? And then we have to decide if it's going north or if it's going south. And then we have to decide which state it goes to. And then we have to decide from that state which county. And then from there which neighborhood. And then from there which street, until finally we get to which address. And basically what happens is, as the proteins go through these different membranous sacs, they're going to get more and more information about where they are finally going to be sent, and then eventually they're going to get packaged into a vesicle that is filled with all of the mail for that particular destination, right? All of the proteins destined for transport to wherever that particular protein is going. And so that's a very important sorting mechanism for the proteins that are going to be created by the ER. Again, a lysosome is a specialized type of vesicle that has digestive enzymes in it. A peroxisome is a very similar type of digestive vesicle, but instead of having digestive enzymes, it actually is going to have oxidative enzymes and catalase enzymes like hydrogen peroxide. And peroxisomes, again, they're called that because they have peroxide in them, are going to come from pre-existing ones. So before they digest themselves or digest something new, they're going to create a new peroxisome. So lysosomes and peroxisomes are very important because they are basically the cellular trash can and the cellular recycling center. Another very important cellular recycling center is a proteasome. Anytime that we have proteins that are either unneeded or they've expired or they've stopped functioning properly or they were created improperly in the first place or they got damaged or something, they're going to go to the proteasome where they're going to be digested into small peptides, which can then be recycled into creation of new proteins. Again, the cell doesn't waste anything that it might need later on. And mitochondria, last but not least, are a specialized type of cell that have a inner and an outer membrane. And that's interesting because this goes along with that endosymbiont hypothesis I talked about previously, which said that the mitochondria and chloroplast too were their own small prokaryotic organisms that got engulfed by a eukaryotic organism and then ended up being able to perform the function of creation of energy. Again, it did that two different ways. The mitochondria create ATP through something called the electron transport chain, and chloroplasts are going to create energy by way of photosynthesis from the energy of the sun, right? So creating of glucose. But either way, these are specialized things that happen only in the specialized organelle type, which actually at some point was its own prokaryotic cell. And we know that because mitochondria and chloroplasts have their own specialized DNA and they create their own new organelles. So mitochondria create new mitochondria and chloroplasts create new chloroplasts. They don't end up dividing when the rest of the cell ends up dividing, right? They just kind of create their own within the cell. But mitochondria, again, they're very important because they create ATP. And then they also are going to play a role in early apoptosis. So apoptosis is programmed cell death, and so they are going to also be involved in orchestrating the early stages of programmed cell death. Now the nucleus, as I mentioned previously, is kind of like the boss's office. It's a very specialized region, and it not only is inside a plasma membrane like all of the rest of the organelles, but it's inside a nuclear envelope, which actually means that it's two layers of phospholipids. So it's a double phospholipid bilayer. And that means that it's able to completely segregate from the inside and the outside. Things are only going to pass into the nucleus through the nuclear pores, so there's no other way of getting in. And the only things that go out are going to be RNA, etc., and the ribosomal RNA, mRNA, but the DNA is going to stay inside the nucleus at all times, right? And it says chromosomes now, but chromosomes are only during division of the cell. Normally, under regular cell conditions, it's going to be a tangled mess of what we call chromatin in interphase cells. And you can picture this, the difference between having a girl's hair up in complex braids or having a girl's hair down and loose so that you're able to actually run your fingers through it. That would be the equivalent of chromatin, which would be what was actually in use, versus chromosomes when it's all up in braids and you can't actually access any of the material. And again, the nuclear pores are the region which are going to allow substances into the nucleus from the cytoplasm and out of the nucleus into the cytoplasm. We also have a specialized region within the nucleus called the nucleoli. Nucleoli are going to be regions that produce ribosomes. Again, the ribosomes are going to get exported out and then be part of protein synthesis. And we also inside have chromosomes. Again, the chromosomes are usually chromatin, but when they're all wound up, they're chromosomes. And they have specialized regions called genes that are involved in controlling everything within the cell. And that's going to get its own chapter. All right, so the cytoskeleton is a very important, it plays a very important role in the structural and scaffolding of the cell. So these are microfilaments, intermediate filaments, and microtubules. The microfilaments are the very smallest ones. You can see those examples here protruding out. They're going to help keep the structure of the cell sound, and they also can go into things like the microvilli when we are talking about, for example, the intestines or different regions that might have microvilli for absorption. And that's the microfilaments are going to be involved in keeping these microvilli erect. We also have these intermediate filaments, which are a little bit larger, and then microtubules, which are much larger, and which are specifically involved in the creation of the spindle fibers, when we get to division of the cell. And that's actually what's being depicted here is the microtubules are coming across and they're actually getting ready to interact with this as a nucleus that is dividing or is about to be dividing, which means that soon it's going to enter into prophase and soon they're going to end up separating from one cell to the other. And that's what microtubules do. They help pull DNA that has already been replicated from the midline of the cell to the opposing poles so that it can then be divided into two cells. But that's not all the microtubules do, right? They're also involved in the flagella and the cilia and the specialized types of cells. All right, so centrosomes and centrioles are very important in cell division specifically and also in the arrangement of a like the flagella, for example. So centrioles actually are going to have these specialized microtubules that are, in this case, they're in triplets, and they're going to be arranged kind of like a little flower structure to help make a very organized region that we can have the spindle fibers attached to be able to pull the chromosomes closer. And we'll talk about that when we get to cell division. Now, I also mentioned that microtubules are involved in cilia and flagella. I did mention that cilia were stationary. For example, they line the trachea, they also line the uterus, and they allow fluid to flow across them. And you can picture this more along the lines of a rowboat or a bunch of oars on a rowboat, as opposed to, in this case, it would be like a one propelling mechanism at the very back, which would be something like the outboard motor. And this, in this case, again, the sperm cell is actually moving through the liquid medium instead of the liquid medium moving across the cells. And if you do a cross section in a cilia or a flagella, it's going to look very similar to what we just saw when we looked at the centrioles, in that we have three microtubules in a line arranged in a kind of a flower pattern here. We also have doublet microtubules and a central pair of microtubules as we move outward. So the basal body is going to look a lot like the centriole that we just saw, that as we head backwards down that flagella or cilia, we're going to end up with doublets instead of triplets, and we have a central one that's going to be picked up as well. And this is going to allow for the motion of the cilia or the flagella, because we're able to pull and contract on one side and release on the other, which is going to allow for the motion of this little tail region. Last but not least, one of the final major differences between cilia and flagella. Now again, the cilia, which is shown here, is going to be stationary and fluid is going to move across it, but basically this is going to whip, right? And it's going to whip the fluid across. It's going to go up and extend and come back down and then go out again, right? If flagella, however, is going to move more like this, kind of like in a sine wave, and it's going to allow again for the motion of the cell as opposed to the motion of the fluid. All right, so let's change gears for a second and talk about ribosomes. Ribosomes are involved in protein assembly. We have a large and a small ribosomal subunit that sit together, kind of like a zipper. And inside here, these will not assemble until we have an mRNA transcript. mRNA is a messenger RNA. Actually, these guys are also made of RNA. So this is a small RNA, small rRNA, so R being ribosomal RNA, and a large ribosomal RNA. And then the messenger RNA is going to fit right in the middle there. The messenger RNA is going to be the transcript, which again is going to come from the nucleus through the nuclear pores to the ribosomes in order to create proteins. And where does that happen? That happens again on the ribosomes, which are either free in solution or found attached here to the rough ER. And this here is depicted as a smooth ER. You can see these are connected by the endomembrane system. And again, they called it the rough versus the smooth because when they looked at the microscope, literally that's what they saw, right? This is the rough ER with all these little dots all over it. This is the smooth ER without any of those little dots, which we now know are ribosomes and are the site of protein synthesis. But don't forget that this is responsible for creating fatty acids in membranous areas, so this is actually going to be the site of phospholipid synthesis as well. So this is not just a useless region, this actually has a whole different purpose. It just is not involved in the creation of proteins. Okay, so once proteins are created, they're exported from the ER to the Golgi apparatus. And as I mentioned, this is kind of like the U.S. Postal Service, right? You have a zip code. In order to get to that zip code, you have to first go through multiple different steps, right? Different hubs. So first we're going to say everybody that's going east goes to this side, everybody going west goes to that side. Okay, cool. So now we have east going one way, west going the other. So now east goes forward, and then we go north or south. If you're east-north or if you're east-south, for example. And this is basically just a way of the cell going through multiple different ways where it gets the protein within the cell going through multiple different layers, whereby it's going to get tags that are going to indicate its final destination within the cell. So it enters in at this cis phase here. It goes through multiple different differentiation steps where it gets told to go left or right or left or right or left or right, and eventually it's going to get secreted out into these little vesicles that are going to exit off the trans phase, and are going to head to whatever their final destination is. And at this point, everything in here is going to that specific, as I mentioned, neighborhood or street. And they can go almost anywhere, right? They can end up going to the plasma membrane, for example. They can end up getting exported by exocytosis, right? So these synthesized proteins can go to a lot of regions. So here, let me depict backwards. Here's the ribosome that we just talked about. Remember the ribosomal RNA? We have a large and a small subunit. We're going to have mRNA through here too, which is not depicted. That creates a protein. Once that protein is synthesized, then it goes into a vesicle and gets transported to the ER. So you can picture this like a baby protein that has no imprinting on it. We have to figure out what its address is. So first we have to figure out what state it's going to. Well, first we have to figure out east, west, north, south, and then figure out what state, and then from there figure out what county, and then from there figure out what street, etc. When it finally is going to reach its little transport vesicle, it is then going to be ready to go towards either, whether it's going to secretion or to the membrane, or to end up being transported to the lysosome, etc. Each of these are going to have final destinations based on where they're going to have their final function, and it will have been sorted out as it goes through the different layers of the Golgi complex. Now, lysosomes, pretty simple to concept. These are going to be vesicles that have digestive enzymes inside. We also have peroxisomes, as I mentioned. Those are going to have peroxides inside. And these are going to be responsible for degradation of different foreign invaders or also misfolded proteins, etc. And they're basically going to be ways in which we can digest the different waste products into what we want to recycle and what we want to secrete. Right, so lysosomes are going to do that by way of digestive enzymes. Peroxisomes do that by way of enzymes that are going to use oxygen to oxidize things, so things like peroxides. And then also proteasomes, another specialized type of recycling container, are going to help destroy any of the proteins that are unneeded, damaged, faulty, etc. by chopping them into smaller and smaller peptides until eventually they get down to their basic amino acid subunits, and then they can be recycled into the creation of new proteins. So these are all ways in which the cell takes things that are not necessarily needed at the time and either breaks them into their original components or digests them and degrades them to be able to send out for waste. Okay. Mitochondria are very specialized. Again, they were originally their own prokaryotic organism that were then digested by a eukaryotic organism, and it was determined that instead of eating them, it was better for the organism to keep them alive because they create energy. So how do they do that? Well, one of the things that's interesting about them that is actually helps to support that endosymbiont hypothesis is that they have two separate membranes. The outer mitochondrial membrane mimics that of the organism that it's in. Right? So if it's a mitochondria from a dog cell, it's going to have a dog membrane. If it's a mitochondria from a human cell, it's going to have a human membrane. And so it's going to mimic the membrane of the organism that is its host. But the inner mitochondrial membrane is going to have specific membrane characteristics of the mitochondria. Additionally, we're also going to have mitochondrial DNA, which is one of the ways you may have heard of the 7 daughters of Eve, that you're able to trace back your maternal lineage. And I say maternal lineage specifically because in that sperm cell, unfortunately, the mitochondria are found in the tail, and so only the head makes it in for fertilization. So all the mitochondria are lost at fertilization. So all of the mitochondria that get passed down come from the egg, which means it travels back through the mother's lineage if you were to look at the DNA. But I digress. The very cool thing about mitochondria is that they have this electron transport chain that happens because of what we call cristae. So there's these folds here, right? And inside these folds, we have these specialized enzymes that help assist with what we call the electron transport chain, and that's going to help create ATP, which is then going to get released back into the cell. So then this way the mitochondria are going to get protection from the external environment by the host cell, and they're also going to help create energy for the host cell. So that's why it's called symbiosis, and by both organisms are going to be benefited by this, as opposed to something like a parasitic relationship whereby only one organism would benefit. But I digress. All right, so let's talk about the nucleus. We talked about this being the boss's chambers, right, the boss's office. And inside, it houses all of the DNA. And normally the DNA is unwound into the chromatin, which just kind of looks like this. You can picture the difference between actually using all of the books in the library. They might be sprawled all over the desk so that you can make Xeroxes and go through them with your markers. I know that's kind of outdated for those of you that are all electronic, but one of the difference between having all of those books put back on the shelf so that the library can take pretty pictures, right, which is the equivalent of having everything put away for chromosomes, right? So when we're ready to divide, all this chromatin is going to get condensed into chromosomes. But right now it's decondensed, and that's important because we're using it to create RNA. So DNA makes RNA, and RNA makes protein. All right? Again, the nucleolus is where we are making the ribosomes. The ribosomal RNA comes from here, and the chromatin makes RNA from DNA, that RNA is going to be secreted out from the nuclear pore. These nuclear pores are depicted here in red, right? They look like little donuts. And the DNA cannot go out and the RNA can. And that's very specific because DNA uses A, T, C, and G. RNA uses a U, C, and G. So that U instead of T is going to be one way by which, say, there's gatekeepers here at the nuclear pore, by which the gatekeepers are going to know that it's going to be allowed out. Another way is that ribose is inherently different from deoxyribose, so the sugar in RNA is different from the sugar in DNA, and that's because deoxyribose, it's written right in there, is actually missing one oxygen compared to ribose, and so that's different as well. And last but not least, RNA is typically going to be single-stranded, and DNA is typically going to be double-stranded. So those three things are going to tell the gatekeepers of the nuclear pore whether or not it's RNA and should be allowed out, or whether or not it's DNA and should not be allowed out. And again, once that RNA comes out of this nuclear pore, then it's going to bind with the ribosome, either free in solution or in a polyribosome unit, which is all around the nucleus as well, or here on the rough ER. Again, the nucleus contains all of the genetic information. When they are arranged in chromosomes for division, we have specific regions that are called genes, and these genes are going to be specific subunits. So we could have gene A, gene B, gene C, right, each in order, and they would be specific to the organism that has the DNA. Let's talk about the structure here. So this whole thing, this entire X, is a chromosome. These are each sister chromatids, right? So they are duplicate. This is exactly even to that, but until they are separated out into two of the actual I's instead of an X, they're considered chromatids. So they're sisters. This here region is the centromere region, this part right in the middle, and inside there's a region called the kinetic core. And that's going to be important when we talk about cellular division, because that's where the microtubules actually are going to bind to the chromosome itself. All right, so this is DNA that is decondensed, right? So this is chromatin. And if we want to condense it down to be able to allow for cellular division to occur, we have to get these chromosome forms. And we do that by multiple sets of condensation, essentially. We wrap the DNA double helix around a histone protein. Each histone, there's actually 8 of them. It becomes a histone octet, which turns into a nice pretty little ball. When we wrap around the histone octet, we do that 2.5 times, 2.5 times, 2.5 times, and in that way we shorten length dramatically. And then we do another set of folding whereby we fold these chromatin that are wrapped around the histones, which then becomes a nucleosome. We put all these nucleosomes together in another set of wrapping, and then we do these loops, which is eventually going to get us to completely condensed coiled DNA. And as you might imagine, your cell is not able to use the DNA while it is compacted like this. So it's only going to look like this when the cell is ready to reproduce, right? When it's just about to divide. But it's a really good way for scientists to determine the location of genes when we have it condensed like this, because we're able to use different fluorescent mapping to pick out which genes are located to specific regions. And now we've mapped the entire human genome because we have the computer power to do so. All right. So as I mentioned previously, DNA makes RNA and RNA makes protein. So what does that look like? We have 2 major steps. The first is what we call transcription, which is when we are taking the DNA and we are making RNA from it. As I mentioned previously, DNA is double-stranded and it uses the sugar deoxyribose. RNA is single-stranded and it uses the sugar ribose. Additionally, it also has a different set of the base pairs that it uses. DNA uses A, T, C, and G. RNA uses A, U, G, and C. So that T to U transition is another reason whereby those gate guards are going to allow information out of that nuclear pore or not, as I mentioned. But basically, after we have created RNA from DNA, so we take a long strand of DNA and we make a copy of it, that's called transcription. And then we export it through the nuclear pore into the cytoplasm, where it's going to bind up with ribosomes and create the protein. That's called translation. And you can picture this like if this was a cooking book that has all of the recipes of the entire planet in it, but you only want to make one recipe, right? Then you would take a specific region of it called a gene, which would be the original recipe. You would make a Xerox of that recipe. That would be the RNA. That Xerox only has that recipe. It doesn't have all of the recipes that the DNA have, which would be the entire chromosome, only that specific recipe. But again, the recipe isn't the final product, right? You actually want cherry cobbler or whatever, so that recipe in the form of RNA has to go out of the nuclear pore and be translated were actually cooked in the oven to be able to make the cherry cobbler, right? And that's the protein itself would be the final function. So DNA makes RNA and RNA makes protein. Again, that's the original cookbook. It's a Xerox of a specific recipe, and then it's the creation of the food that's in that recipe. So we have, again, two major steps. Transcription is the first major step. Transcription is when we are creating RNA from DNA. And the way that we do that is we have a specific region of the DNA. In this case, this is a gene, right? Upstream of that gene, we have a promoter region. And downstream, we have a terminator region. Again, you only want to make a Xerox of the specific recipe. You don't need 3 recipes. So you're going to start somewhere, which is the promoter region. You're going to continue to make your Xerox until you get to the terminator region. And to do that, we use a specific enzyme called polymerase, and that's because it makes an RNA polymer. So it's an RNA polymerase making RNA polymer, bringing in nucleotides that were free in solution to bind to the growing RNA chain. Now remember, we're going to use the template from the DNA to be able to create it. So everywhere we have an A, right, in the DNA, we're going to have a U in the RNA, a C in the DNA, a G in the RNA. So you can see that we're using this strand as a template to be able to create this RNA strand. All right, we also are going to have something called intron-exon splicing, which you can picture this like if you were to record an entire TV channel or an entire TV episode, but you wanted to eliminate the commercials. This would be like if you were to stop and snip out every commercial so that you only got the full-length program, right? And then eventually you're going to end up with an RNA transcript, which has all the edits created and already taken out, and then it's going to be exported out of the nucleus into the cytoplasm, where the process of translation is going to occur. Translation occurs, it says it occurs in the nucleus, but it does not occur in the nucleus. I apologize, this occurs in the cytoplasm. That's an error here. Translation occurs in the cytoplasm, and it's basically when we are taking the mRNA nucleotide sequence and using that to translate it into the protein sequence. And so here we have the large and the small ribosomal subunits. Again, these are RNA, so they are ribosomal RNA. And we have the mRNA, or the messenger RNA, which is going to lay right in the middle. In the top, the large subunit, we have 3 regions. We have an A site, which is where things are going to come in, a P site, which is the middle region, and an E site, which is an exit site. So basically you can picture that the tRNA will bind here, here, and here. And I know I haven't discussed tRNA yet. I will in just a minute. tRNA stands for transfer RNA, and these guys are basically the gophers that are going to basically go out into the cytoplasm and find whatever amino acid you need to bring into this growing polypeptide chain to be able to add it. So they're the gophers that are going to one at a time go grab the amino acids that you need, add them to the chain, exit, and then do this over and over again. Now I know there's a lot going on here. Don't worry, we are not going to quiz you on all of the specific details of protein synthesis, but I just kind of want to demonstrate what is happening here. So again, what we are looking at in purple is a small ribosomal subunit. In blue, pink is a large ribosomal subunit. This red line here is the mRNA, or the messenger RNA. And basically the messenger RNA is going to bind to the small subunit, and the large subunit is going to pop right on down. And then we're going to have the binding of the tRNA. And I mentioned tRNA were gophers, but I didn't tell you how they work. So that means that they go out and they have this amino acid on them, but how do they know which one to bring in, right? So the tRNAs have anticodons that are specific to the codon region in the mRNA. So this is important, right? So every time that we have a specific codon here, the anticodon there will bind, and it's a very specific lock and key mechanism. In this case, let's say it's correct. So now that red amino acid is going to be in the A site with this tRNA. This tRNA will transfer the green amino acid over to, and now we've created a peptide bond. We've released the green amino acid, and the green and red amino acid are now a polypeptide chain, or the beginnings of a polypeptide chain. Now, everything's going to shuffle over one in terms of the mRNA and the rRNA. That means that the tRNA is going to get shuffled this direction to the left. This one's going to exit. This one is now in the middle stage. And now a third one can come in to this region to bind. And eventually we're going to end up with a long polypeptide chain, which will hit a stop codon. When we do hit a stop codon, it will be released. And so if you look at this as one mRNA chain, one mRNA molecule, you can see that we can have multiple sets of ribosomes, large and small ribosomal subunits, attached at any given time. So we can use one transcript to make multiple proteins that are all going to be at different stages of being created. As you can see, this one is going to have more on there than that one because we've moved along until eventually they hit their stop codon and the completed protein is released. And then that RNA, that large and small subunit, will find another mRNA transcript to lather, rinse, repeat. Okay, so I just talked about protein synthesis. Now we're going to talk about cell division, and I know we throw a lot of information at you in this lecture, and that's because we really do want to jump into the heart of the matter, and this is all kind of considered background information. But cell division is a very important way by which cells are going to reproduce themselves. Now all cells reproduce themselves, your skin cells, your hair cells, right? And basically what happens is the cell is going to undergo some sort of growth, right, and duplication of all of the organelles and the centrosomes, etc. And then we have to duplicate the DNA. That's called S phase. And after that, we have another set of growth phase. So we have growth phase 1, S phase or replication phase, and growth phase 2, where we're continuing cell growth, and different things are synthesized. All of this is called interphase. Interphase is everything that happens up until the mitotic phase, and this is M phase. The mitotic phase is split up into 3 or 4 different regions. I apologize. Prophase is when we are setting the stage. Things are getting ready to divide. All of our chromosomes are getting condensed. The nuclear membrane is breaking down. Metaphase, we have all of our chromosomes lined up as pretty little X's in what's called a metaphase spread. That means they're right at the middle of the cell and they're oriented all in a pretty little row. This is the point when the spindle fibers are coming off of the centrioles. The centrioles are located at both the left and the right, or up and down. They're at opposite sides of the cell, basically, and they are coming together to be able to meet at the kinetochore region, which is at the centromere or the center of that chromosome, so that they're able to start pulling them apart. And by anaphase, we have actually started to separate out those X's into little L's. Telophase is the process of reconstructing everything that we broke down previously. So telophase and cytokinesis are hand in hand. Telophase is when we're going to start recreating the nuclear envelope, for example. We also, cytokinesis is the tightening of the belt. Basically, it's cell division. We're going to go from a large oval-shaped cell, which has double the DNA, and now we've pulled everything to 2 separate poles, and now we're actually going to split it off into 2 regular-sized cells that each have all of the genetic material. There's also a way out of this that's called G0, and there's a lot of cells in our body that have reached their final destination and they're no longer going to be dividing, and they're considered to be in G0. Eventually they can enter into what's called senescence, but this is just basically like an active cell that is not necessarily needing to replicate and divide. If it does end up in senescence, that's actually kind of a negative term. That means that the cell has lower function or reduced function than it used to when it was a normal cell. All right, so interphase is everything that leads up to the process of mitosis. Again, mitosis is when we are splitting the cell into 2 cells. So we have G1, which is the growth phase, S, which is the synthesis phase, and G2, which is another growth phase, and there's several checkpoints along the way to make sure that everything is a go in terms of the replication of the DNA, and that the cell is chosen to be able to be the one that they do want to replicate. And what has to happen during this region, during this time frame, is that we're going to be growing the cell. We're going to be replicating the DNA, we're going to be doubling all the enzymes, etc. And also here, these are the centrioles. The centrioles are going to be going to opposite poles. Right, but basically what's happening here is that what a normal cell looks like under interphase conditions, which means that you're not seeing the DNA condensed, for example. You can still see, again, we've got chromatin and not chromosomes. Now, during the mitotic phase, we're going to enter into what's called prophase first. Prophase is the very first stage, and this is when we see what was chromatin being condensed into what looks like chromosomes. And that's kind of like an intermediate phase here, where we are in the process of condensing, and then we get to the point where we have condensed. The other thing that happens is that we are losing this nuclear envelope. So previously you saw a nucleus, now you do not see a nucleus. Okay. Additionally, these guys here, which are the centrioles, are going to be moving to opposite sides here, right? And creating these microtubules, which are known as spindle fibers, which are going to be trying to attach to the condensed chromosomes here. So the condensed chromosomes are shown as X's. They have not yet reached what we call a metaphase spread. So prophase is getting ready for the show, right? We are breaking down our nuclear envelope, we are getting our chromosomes condensed, we are moving our centrioles to opposite sides, creating our mitotic spindles, but we haven't actually started the process of anything yet. The next stage is metaphase. Metaphase is when the action actually starts. During metaphase, we have what's called a metaphase plate. So all of our chromosomes are going to line right up in this metaphase plate. Our centrioles are going to be on opposite sides, and this is what this actually looks like if you're looking at a cell that's in the process of dividing that's at its metaphase plate. So these are the centrioles that are going to be having all these spindle fibers coming out. Again, the spindle fibers are going to meet here. This is called the centromere, but there's a specialized region of the centromere called the kinetochore where they're going to connect. All right, as we are pulling the chromosomes to one side or the other, so what was just an X 1 second ago is now going to become little I's, right, or L's. And they're going to get pulled from the spindle fibers towards the centrioles, all right? And that happens during early and late anaphase. So during early anaphase, you can see they're closer to the metaphase plate, right? Here they were in the plate, but they're pulling apart. Now they are getting to the point where they're separating. This is almost telophase, so they are being pulled to opposite sides. This is developing here. This is the cleavage furrow, which is basically the precursor to what we're going to call cytokinesis, which we're going to kind of like a tightening of the belt, where we're going to go from one long oval to like a peanut to two little globes, right? So the cells are going to separate out. Telophase is that last step. Telophase and cytokinesis kind of go hand in hand. So if you picture telophase as like walking out the door and cytokinesis is the door shutting behind you, they kind of happen at the same time. So telophase is basically when we're going to have the dissolution of the mitotic spindles, because we've already brought the chromosomes over to the regions where they're going to be residing. And we're also going to be reforming this nuclear envelope. Right? Chromosomes are going to start to decondense, so they no longer look like little lines. Sorry, they're starting to dissolve back out into chromatin. And that cleavage furrow is going to continue down until we end up separating them out into two, which is again the process of cytokinesis. Cytokinesis is the process of this cleavage furrow here, which currently, remember, first it's going to look like an oval, then the oval is going to turn into a little bit of a peanut, then the peanut is going to turn into a bigger peanut, and then eventually turn into two separate identical cells. And then once that's finished, right, cytokinesis is the end of the mitotic phase, and we're going to go back into what's called interphase, right, which is the time frame when we are deciding whether or not to divide. This is an overview of all of the events of the somatic cell cycle. I have talked about them extensively, so I'm going to skip this, but if you need to refer back to them, this is just kind of an overview of what I just told you. Now let's talk about cell fate, right? So cells have multiple different types of destinies. The first would be something like a stem cell, which is going to remain alive. I apologize. This is more like a cell that has entered into G0. It's going to remain alive and function without dividing. So it's going to continuously survive, but never create more. You can picture this like nervous tissue or other types of fully differentiated tissues. Or it can grow and divide, right, and make more cells over and over, or it can enter into cellular senescence, which is basically cells that have gotten kind of old and are losing their function, or into apoptotic conditions where cell death is going to occur. So either you remain alive and you do your function and you don't make any more offspring, or you continue to grow and divide, or you enter into a cell death pathway. Now let's talk about meiosis. So meiosis is going to be a specialized type of cell division where we are making sex cells. That means we're making sperm cells and we're making egg cells. Now, meiosis is actually really important because it's going to give rise to genetically unique offspring. And what that means is that we're going to, each the male and the female are going to be creating cells that have half of the genetic material. They're called haploid. So instead of having 2 copies of every chromosome, they're going to have 1 copy of every chromosome. And then they're going to recombine with the mate's set of haploid material so that then they can create a new diploid, so again 2 copies of the chromosome, that is completely genetically different from either a parent and also from siblings. Now, another thing that happens during meiosis that's worth talking about, so what we were just talking about was independent assortment, which is how different chromosomes are going to separate out during the process of meiosis, but another thing that happens is something called crossing over. So sister chromatids are going to align next to each other during meiosis, and then they are going to have an area where they're going to switch all of the genes downstream. And so in this case, we have one allele that's one chromosome that has the lowercase alleles and the uppercase alleles from here down. And then the reverse is here, has the uppercase alleles and the lowercase alleles from here down. I want to make a point that what we're switching here is still hair color for hair color, eye color for eye color. Okay, we are not switching different genes around. We're switching the same genes but different versions of those genes. So it's still eye color for eye color, but you might switch blue eyes for brown eyes, for example. And that's called genetic recombination, and that's basically a shuffling of the material before you go ahead and create the sperm and the eggs. Now, meiosis has two different steps. So meiosis 1 is very similar to mitosis 1 with the exception of that crossing over. And then meiosis 2 is going to be a separate set of all of the same stages, right? Prophase, metaphase, anaphase, and telophase, except the end goal is to create haploid cells. Again, haploid means that you have one copy of every chromosome instead of two, and that you have to interact with another cell that has one copy of each to be able to restore the full genetic complement to make the diploid individual. This is why a sperm and an egg have to come together to be able to become fertilized. All right, so we just talked about all the different types of cell fate, right? But we didn't talk about cell diversity. And there's so many different types of cells within your body that all have very specialized functions, all have very specialized forms. They look completely different, and they're utilizing different genes. So although they all have the same genetic material inside their nucleus, different genes are turned on and off in different cells, giving them rise to different cellular functions, and again, different cellular forms, right? So this is a nervous cell, which can be feet long, right, if it's running from your brain all the way down to your toes. Whereas red blood cells are some of the smallest cells in the body, and they are also anucleated. They actually don't have a nucleus at all. They're created by bone marrow and recycle through your body every 60 days or so. All right, so we're going to end our lecture talking about what happens to our cells as we age. And I talked about cellular senescence, which is basically the cell's loss of function. And all of our cells in our body can only be copied so many times. So every time that you make a mitotic copy, you're making a Xerox of a Xerox of a Xerox of a Xerox, right? And so the older that we get, our Xeroxes have been copied more and more and more times. So our cells are gradually going to start to deteriorate in their ability to function and to respond to environmental stresses, etc. We also end up with atrophy of our cells and of our muscles. So the number of our body cells actually decreases over time, and our extracellular components are actually going to get broken down as well. So as we age, our cells are going to lose a lot of their function, and so it's very important that you maintain a healthy environment and you exercise and you eat a healthy diet, etc. Speaking of healthy diets, things that are like free radicals, which I may have mentioned in a previous class, can exacerbate this process. And free radicals are essentially going to be ions that are going to steal electrons from other atoms around them, which then causes the chaos that I mentioned about everyone stealing one pencil from another and no one taking notes. So that's something that actually accumulates over time as we age as well. So I do appreciate you giving me your attention for this lecture, and I hope to see you all again very shortly. Happy studying!